welcome to our July Roundtable. And it's really good to see so many of you here. I am going to present Claire, and she's the main act today. And unfortunately, today we won't have the student talk preceding the main talk, but I would like to use this opportunity to encourage all the students and postdocs who want to present at our roundtables, the 5-minute slot that we usually have, to please come forward and present at the next meeting. So it would be really good to see someone from our hub presenting alongside our big speakers. So today it's Claire O'Callaghan, and she's going to be talking about the window into noradrenaline's role in decision making and learning. And that's quite on point because we used to be the decision science hub. So I think that your talk is going to cater to both our decision science members and the cognitive neuroscience members. Okay. In her own words, Claire is trying to, she's leading a group, slowly trying to, sometimes successfully, do interdisciplinary neuroscience. I think that they're slowly trying, and they sometimes successfully is speaking to the character of Claire being modest, but she is a senior research fellow at the University of Sydney, and she did a postdoc in the University of Cambridge and then returned to Sydney and now leading her own group. So without further ado, I'd like to welcome Claire.
Thank you. Thanks, everybody. Thanks so much for the invitation. It's so lovely to come down here, and I really love that you have this this kind of hub of cognitive neuroscience. We don't have this at University of Sydney. Everybody's spread out, so it's great to see you get it together. And yeah, so in the spirit of cognitive neuroscience, I've tried to talk about some stuff that has a real cognitive focus. So hopefully it's interesting for people. It actually has quite a clinical bent, some of this. So we'll see how we go. And then, yeah, at the end, I thought to try flag a couple of other directions that my group and my colleagues are working on, just to, because I think it's so often in Australia, we are a little bit siloed and we don't know what other people are working on. So I tried to do a little bit of that at the end, but we'll see how we go. I'll try to leave a good chunk of time for questions. I always have too many slides. So if you sort of notice me speeding up a little bit, it's because I'm trying to make time for questions. Cool. All right. So yeah, I never know where to start this kind of talk. Today I'm going to start by talking a little about the noradrenergic system, which is a system that I love very much. Yeah, it's this beautiful distributed neuromodulatory system in the brain, emanating for the most part from this tiny nucleus in the brainstem, the locus coeruleus, which in humans is really, really tiny. It's sort of about 1 millimeter by 1.6 millimeters deep in the brainstem, nestled right next to the fourth ventricle. So people that do neuroimaging will already know that that's a very challenging part of the brain to think about for imaging, particularly functional imaging. But it has all these, sort of remarkable properties. It projects obviously to many more places than the cerebral cortex, but it has these sort of different modes of modulation. It can sort of act in this more targeted mode where these sort of ensembles of noradrenergic neurons sort of get together and target particular bits of the brain. But it can also kind of really sort of slam it on in this more sort of global broadcast mode where it can sort of have these far-reaching projections and very sort of large impacts on brain and behaviour. And another little bit of it that sort of adds. So that should be a brain there. It's not rendering quite well, but imagine a nice looking brain there. Yeah, so another sort of part of the system that adds nuance is that it has these different receptor families that are distributed all over the brain, and how noradrenergic modulation affects those receptors really depends on sort of where they're situated, whether they're pre or postsynaptic, whether they're in the front part of the brain or the back part of the brain. So it's sort of this, you can kind of think of it as this system that sort of is so widespread throughout the brain. When we think of the other kind of classic neuromodulators, probably only serotonin is sort of equal in sort of how widely distributed it is. Things like dopamine and acetylcholine are much more sort of targeted in their projections. So it's super widespread, so it can have these really wide-ranging effects, but it has all these mechanisms to it that allow it to have much more nuanced effects as well. So it's a beautiful system for sort of thinking about sort of controlling adaptive behaviour. The other super cool thing about it is that it receives descending projections from most parts of the brain. And yeah, they actually land in this little territory, not necessarily in the locus coeruleus itself. Oh, you'll see this little red kind of little like two red sausages poking up often in the talk. And essentially that's the sort of shape. If you were looking at my brain sort of front on, the locus coeruleus would look like that in humans, this sort of elongated structure. So that's sort of how we render it in humans. But yeah, these descending projections, they sort of hit this this little periceruleus region where there's this GABAergic sort of family of neurons that do all this sort of inhibitory regulation over the locus coeruleus proper. So it's this amazing sort of multi-leveled system that's modulating the brain, but then being modulated by the sort of very parts that it itself targets. And then this sort of, really fascinating sort of microcircuitry going on at the level of the locus coeruleus where there's this sort of inhibitory regulation. So yeah, of course you can imagine this beautiful system is so well placed to kind of, modulate or toggle adaptive behavior. And I like to think of it, I guess, at sort of, 3 levels. You can kind of think of it at the level of how these cells, how the locus coeruleus noradrenergic cells behave. And like many cells in the brain, they have these different modes of firing. So sort of phasic modes and tonic modes of firing. And yeah, essentially the sort of relative balance of when the locus coeruleus is sort of firing off in a phasic way versus tonic firing has different effects on behavioral engagement. And one particularly sort of famous dichotomy is that, when you've got sort of a moderate level of tonic firing and phasic firing, you're pretty task focused. You can kind of exploit what you're doing and pay attention to things that you care about. But when those tonic levels rise, that causes sort of task engagement, which on the one hand, you can think of, oh, maybe that's going to make me sort of perform crappy at what I'm doing. But actually, you can also think of it as the thing that lets you leave off from the task that you're doing and focus on something else. So it's a really beautiful driver of sort of engagement with your environment. And then you can sort of think about more at the kind of network level, the sort of effects it can have, I guess, on large scale brain networks, which is something that people, in sort of fMRI research are interested in, but even sort of, more sort of smaller circuits, that it can have these kind of effects of sort of, injecting noradrenaline into networks, which allow a network to reconfigure. And the reason that the little crab is there is that these ideas are based on sort of original work where, these sort of models in crustaceans where you'd sort of have, like a pattern generator or something with not even that many neurons. But as soon as you kind of squirt a little bit of neuromodulation into even a sort of relatively simple network, it can completely reconfigure it and let it sort of reach all these new kind of heights and sort of, I guess, dynamic forms that it couldn't do without neuromodulation. And then I guess the last level to think about it is, kind of what does this system care about in the environment? What kind of, triggers it? And it's definitely something that, it's paying attention to sort of, changes in your environment. So if you're sort of chugging along and things are the same, your locus coeruleus is, probably pretty quiet. But, when something happens that's worth paying attention to or worth learning, your locus coeruleus is going to speak up. So it's easy to imagine from there, if you think about kind of the different behaviors that it, I'm hoping some people appreciated the motor, thank you. And yeah, so it's easy to think about, if you think about all these adaptive behaviors that it's involved in, it's easy to sort of think, how would these map to symptoms in neuropsychiatric illnesses? And the type of, in my group, we sort of tried to kind of have a balance of doing more, I guess, basic science, sort of blue sky work, and just trying to understand how the brain works, and also having a kind of clinical arm. And yeah, for us, the sort of clinical conditions that we're interested in are these sort of neurodegenerative or neuropsychiatric diseases of aging. But certainly it goes without saying that in a lot of developmental and psychiatric illnesses, the locus coeruleus noradrenergic system is a big player as well. But yeah, some of the kind of symptoms that you can see that, these I guess, behavioral correlates of the locus coeruleus noradrenergic system, some of the things that they map onto that are really interesting for clinical populations is this sort of, I guess this sort of spectrum of, engagement versus disengagement with the environment, and also things to do with kind of learning and memory. It modulates learning and memory on many different levels, but I guess at one level, it's even just this idea of kind of knowing what to care about in your environment. So it can be sort of attended to, and then sort of, committed to memory. So I guess, yeah, I already sort of flagged flag the idea that, yeah, we try to sort of work on problems that are more just, yeah, trying to understand how the brain works, but also trying to see if we can come up with some interesting clinical problems. And I think it's funny, and I guess a lot of people here, if you work in cognitive science or cognitive neuroscience, if you do sort of clinical work, some of it does have a very, let's say, translatable thrust to it, that, you know, you can really see it sort of helping or solving a clinical problem. But then some of the work is also just using these sort of clinical disorders as models to help us understand behavior. And I like to think, at least I guess some of the work that I'll talk about here, I like to think it has a bit of a flavor of both, that it's sort of answering some interesting, some useful clinical questions, but maybe it gives us a bit of insight into what this system does. And I guess, yeah, one way to kind of think about it, you know, I think a lot of what we do in sort of cognitive neuroscience is, you know, at this level here, sort of working with animal models or healthy humans, you know, sort of undergraduates, you know, locked away in the lab doing hundreds of trials of some kind of task. And in this way, you know, this captures something pretty close to how behavior works. You can imagine, you know, a kind of orchid, it's, you know, it's growing, it's pretty natural, but, you know, it's still in a pot, still constrained a little by these, you know, little pegs. So it's not completely natural. And, you know, another way that we can come at things, you know, try to understand behavior in cognitive neuroscience or computational neuroscience is, you know, building biophysical models. And this is, you know, for the people that, you know, don't want to waste time collecting data, and you can build something, you know, that looks just as good as what you'd collect, and is, you know, far cheaper. You can get it at IKEA. And yeah, I'm not in any way trying to discredit that type of work, because yeah, a lot of my colleagues, we work together on those ideas. But I guess it's sort of this sort of, you know, you're kind of capturing the shape of something, but, you know, it's still in this quite unnatural way. But then I guess at the other extreme is what you can learn from patient work. And it's almost like imagining this system or this sort of function that you're interested in, but how it's completely out in the wild, you know, all of these kind of things that, you know, it comes with all of these sort of caveats of other things you have to consider, you know, other sort of factors. The fact that something like Parkinson's is a massively multi-system disorder. It affects your whole brain and body. But, you know, arguably you're still capturing sort of something in a very, very natural way, just with a particular set of caveats, in the same way that you're capturing something beautiful and important with biophysical models, but the set of caveats is different. So yeah, as I mentioned, I'll zone in a little bit on the locus coeruleus noradrenergic system in Parkinson's disease. And this is just from a recent review paper we worked on, led by one of my students, which is a lovely paper. To credit her, not me. But yeah, in Parkinson's, as in many of these neurodegenerative diseases of aging, you see a real sort of suite of different sort of pathological changes at the level of the locus coeruleus. You know, you can kind of, you probably all sort of imagine, you know, cell death, sort of, you know, cell loss, but, you know, you can, I guess, imagine these kind of phases of sort of pathological accumulation, cell loss, but also kind of compensatory processes. You know, you see axonal sprouting and that sort of thing. And, you know, the functional consequences of this are really complex. You know, again, we sort of like to imagine simple trajectories in these kind of illnesses. You know, you get an illness and stuff goes bad and it just keeps going bad. But, you know, actually the way sort of cells and circuits are functioning, and consequently behavior are functioning, is on a very non-linear trajectory, often with sort of, increased function, attempts at compensation, and then sort of, you know, cell death and sort of a real sort of mishmash of how things go. And I won't dwell on this too much, because I guess that's a topic for another day. But I guess just to sort of, just to kind of encourage you to think about, when we kind of imagine targeting these illnesses with drugs, it's very complicated, because it's such a nonlinear process and such an individualistic process of how these systems change. And this is just one system, you know, compared to sort of all the others that are going wrong at the same time. And yeah, locus coeruleus noradrenergic pathology in Parkinson's and in other diseases of aging, like Alzheimer's and some of the frontotemporal dementias, they're associated with a variety of symptoms. And I guess it's not hard to imagine, you know, because it does have these widespread projections, and I've not even mentioned its sort of descending projections to sort of other lower brainstem nuclei that regulate autonomic function and sleep and things like that. So yeah, when this system goes wrong in any condition, it a whole gamut of things go wrong. But, you know, being a cognitive neuroscience hub, we're going to sort of think about some of those top ones, like cognition and neuropsychiatric features. And a super important thing to notice, and this goes for Parkinson's and Alzheimer's, is that the locus coeruleus is one of the earliest sites of change in these illnesses. That sort of staging scheme essentially refers to the fact that when you're at this stage 1 or stage 2, you wouldn't even know that you've really got the illness because it's sort of typically not presenting in a way that's sort of manifesting in discernible symptoms, and often there's a lot of compensation going on at that time. But surely enough, from the earlier stages in things like Parkinson's and Alzheimer's, we see already quite widespread changes in the locus coeruleus noradrenergic system. So given that I've told you sort of how complex and interesting this system is, and focused on how it changes in a particular disease, you can sort of already imagine it's a kind of hard thing to get at and to study, and particularly if you want to kind of study it with a view to ideas of what might be useful for people with these illnesses. So an approach that we took, and this is work that I did in Cambridge during my postdoc, and work that we're trying very hard to set off here, but it's much slower, but getting there. But it's this idea of sort of a single dose drug study approach, which is actually a sort of study design that I really love. It's the idea of kind of giving people a one-off dose of a drug, and if you like, chucking them in a scanner, measuring what their brain's doing, or getting them to do behavioural tasks. And I think it's in diseases, I think it has a very interesting clinical application, which hopefully will become evident. But even just as a way to sort of study healthy human cognition, to try to understand what these neuromodulatory systems are doing, using these drugs as sort of a perturbation to sort of, get more knowledge of the system. So yeah, I'm a big fan of these study designs, even though they're quite challenging to do. But what we did in this particular study, so we were quite interested in people's behaviour. So we didn't sort of want to scan them on and off the drug, these patients with Parkinson's disease, but we did collect data, some baseline scanning data, because to preempt things I'm going to talk about, we were very interested in the question of whether the sort of integrity of people's locus coeruleus noradrenergic system, whether that might relate to how they responded to a drug, right? And this gets to a really, kind of interesting fundamental principle of neuromodulatory systems. I've already given you, I guess, a flavour of sort of how complex and wide-ranging the noradrenergic system is. But the other sort of factor is that so much of kind of, I guess, how the noradrenergic system and these other systems respond to the environment, and in turn how they respond to drugs, so much of it depends on people's sort of baseline levels of function, right? So if you're somebody that sort of naturally has a sort of, I don't know, higher kind of dopaminergic turnover or lower dopaminergic turnover, if we give you cognitive tasks, that might kind of alter how you perform on it. But perhaps, most importantly for clinical applications, it's going to sort of change, how you respond to a drug. And I think this is probably something that is, if we imagine kind of personalized medicine in neuropsychiatric illnesses or neurodegenerative, or, symptomatic treatment for neurodegenerative diseases of aging, I think this is kind of one of the, really sort of underdone aspects of it, because essentially, we know a drug works, you just sort of give it to people and you try, but it's going to work in, a few people, not in everyone. And, we need sort of more avenues to think about these sort of personalized avenues of how we best treat people with these illnesses. So that sort of waffled on a little bit and preempted, but yeah, our idea was that, LC imaging, so knowing sort of the state of somebody's locus coeruleus noradrenergic system, will that actually help for, with a view to sort of personalized medicine or stratifying people for noradrenergic therapy. So a little quick interlude about locus coeruleus imaging. And, this goes actually for any of these sort of, imaging these sort of, neuromodulatory systems and any of the brainstem nuclei is very, very hard. So this is imagine sort of looking at a brainstem kind of frontal. There's a lot going on there, as you can see. It's a small area. Things are really, really tightly packed together. And as I mentioned as well at the beginning, a lot of what's going on in the brainstem is, really just adjacent to the fourth ventricle. And even though I'm not talking about functional MRI in this talk, it is something we work on as well in the brainstem. And, if you've ever looked at a functional scan anywhere around the brainstem, it's just, banging like this because there's so much pulsation and breathing and pulsation from the fourth ventricle. So it's super tricky to sort of, it's a tricky area to get at in humans. And in fact, the locus coeruleus is especially hard because one really interesting way to think about these sort of subcortical and brainstem nuclei, I think when we imagine them, a lot of them, we think of these sort of tightly bounded nuclei. You think of the caudate or something like that, something that's got like a nice boundary to it. But there's actually a kind of mix of nuclei, deep in our brain, and some of them have this lovely bounded structure, which is, I think, a bit easier to sort of locate and get at. But some of them are these sort of unbounded, almost like just a sort of smear of cells. And the locus coeruleus, I think, is an ideal one of those, which is captured very well here. It's not a sort of tightly bounded nucleus, despite the lovely red pictures I've put of it. In reality, it's this kind of, this sort of spread of noradrenergic neurons, which in their core are quite densely packed, and then they sort of fan out and get a bit sparser as you go down in the brain. So it's a super challenging thing to try to get at in human diseases or in humans in general. And I think that's why, particularly in the kind of noradrenergic field and locus coeruleus and other sort of small nuclei, there's been, I guess, a bit of a surge of work and excitement, at least from the imaging perspective in recent years, because we're getting a bit better at being able to at least convince ourselves that we might be capturing these nuclei in a sensible way. The cool thing about noradrenaline, it's got this little this little helper that is useful for imaging for us. For the type of imaging that I'm going to talk about, which is not functional imaging, but it's rather capturing the sort of integrity of this system. And this little kind of helper that we have in the locus coeruleus, it's found, it's neuromelanin. It's found in the locus coeruleus and the nigra, so in catecholaminergic nuclei. It's essentially this kind of like chunky little macromolecule that is dark, and that's why these nuclei look dark if you would open up the brain. And it's something that just kind of builds up in your brain over the lifespan. It's insoluble, so it just stays there. So essentially, you don't really start off with any of it when you're young, and over life you end up with a big chunk of it in sort of each catecholaminergic cell in this territory, and sort of by older years, it's kind of taking up half of each cell. And I know that probably sounds bad, but it's actually helping you. It's doing good things. It's not all, but it's funny. It's this thing. I mean, I'm not a sort of, molecular person, and I tried to understand neuromelanin, but there's a big debate about it, still, because essentially it's this. It seems to be this thing that sort of throughout life, it is helping. So what it's kind of doing is, within these, really big catecholaminergic cells that are so important because they're so active, they're, driving dopaminergic, noradrenergic cells are, hopefully, driving sort of everything that we do day to day, and it kind of sort of sequesters, bad things within those cells. So it kind of is kind of like gobbling up and keeping in these sort of bad things in those cells, and that's why it gets bigger and bigger. But then in fact so then it's a whole other thing though when people get these diseases of aging, so Parkinson's and Alzheimer's, and that's when neuromelanin sort of seems to turn nasty and does things that you don't want it to do. But for our purposes, it's great because essentially, people with a more sort of MRI physics background will appreciate it. But as we know from MRI, different sort of substances and things in the brain allow us to use different contrasts and pick up different things. So there's actually a really well developed sequence now. It's a magnetization transfer sequence that picks up the contrast of these neuromelanin cells. And there's a whole other debate about whether it's actually picking up neuromelanin or whether it's, the fact that these cells are kind of large and have lipids. But anyway, it does sort of, it's a surrogate measure for, I guess, the amount of sort of neuromelanin in the locus coeruleus, which then you just need to kind of think one more step down the road. That's a surrogate measure for sort of the, I guess, health of your, how many noradrenergic cells you have left. So we can imagine that when people have Alzheimer's or Parkinson's, they have a reduced contrast on this kind of imaging. So yes, it's very hard, but it's actually doable. And this is just to show a quick little a flash of the fact that we can actually do it at 17, we can see it. And of course I present, a lovely group average, so it looks, really impressive, but I don't know, it still kind of blows my mind to think that we can capture. I mean, just remember it's 1 millimeter by, 1.6 millimeter. That's kind of cool, right? So yeah, we did work on some of that kind of imaging, and yeah, we created an atlas for it, which is openly available if people want to use it. But I guess just a little nod to the fact that it's still a bit of a wild west. People don't have atlases that they agree on, because it is such a hard thing to capture. And like many parts of neuroscience, a lot of how you capture it or what you capture is dependent on the technique that you're applying to it. So ours is the most recent atlas using 7 Tesla. For the purpose of this talk, we can take that as the ground truth, correct atlas, and just go from there. But of course, it's honestly, it's I mean, you can kind of imagine, again, people, if they're using kind of more sort of fMRI and cortical imaging, all the debates that rage about different parcellation schemes, and how to separate bits of the brain. I mean, it's the same question in the same debates, just in a smaller bit of real estate. But actually matters more, actually, in the, if you're trying to, isolate something in the cortex or a big structure, or parcellating something, if you're a few millimeters out, it actually doesn't matter. But if you're a few millimeters out in the locus coeruleus, you're missing it. So it's tricky. But suffice to say that using this technique, so this neuromelanin sensitive imaging, it does let us localize really nicely sort of the I guess integrity of the locus coeruleus in Parkinson's. And we and others have seen this kind of even at the level of a kind of rostrocaudal gradient, where the caudal, that's the bottom bit of the locus coeruleus, is sort of lost more integrity compared to the rostral. And it's actually an opposite pattern in Alzheimer's disease. So that's cool, we can measure it. And yeah, so coming back to the question, yeah, can LC imaging help with personalized medicine? And I guess something that I already preempted and already realized, I probably need to rush through my slides to get to some more results. But yeah, the idea that when you give people drugs, you sort of think everybody's going to get better, but in reality, what happens is a mix of things. Some people get better on a drug, some people get worse, some people stay the same. And yeah, one of our ideas was that, perhaps sort of knowing the baseline integrity of people's neurogenetic system, using this neuromelanin imaging as a surrogate might actually help us to know, what sort of who might be suitable for neurogenetic therapy. So yeah, we used a drug called atomoxetine, which is a great drug. It's been licensed a long time for a second-line treatment in sort of ADHD and developmental disorders. And yeah, part of the idea of this sort of, I guess, clinical bent of the work that we do is the idea of repurposing drugs, because essentially, if you want to invent a new drug, that's sort of, reinventing the wheel, and it's, decades long. But, we have loads of good drugs that are well used and licensed, so we know are safe, that just haven't been applied to things like Alzheimer's and Parkinson's, despite the fact that we've known for a long time just how sort of impaired the neurogenetic system is in these diseases. People aren't routinely prescribed neurogenergic drugs. And yeah, atomoxetine is lovely. It's a noradrenaline reuptake inhibitor, so you can see that it blocks, it sort of blocks reuptake at the synapse, leaving more available in the synaptic cleft. And yeah, it has all these sort of interesting sort of properties where at the level of the locus coeruleus, you sort of see this auto-inhibition, which kind of serves to dampen the kind of, I guess, level of firing, but preserve the phasic. So you get this kind of increased signal to noise throughout the brain. It also increases extracellular availability of noradrenaline. And something to note that it also increases availability of dopamine. And that's something that it doesn't necessarily matter for, the question of, do these drugs help people? But actually, when we think about more sort of, I guess, theoretical interpretations, it is worth noting that, like all psychoactive drugs, noradrenaline, atomoxetine is messy in its targets. It's sort of raising noradrenaline, but also in some parts of the brain raising dopamine. And I think that's, I don't know, a really kind of useful point. I mean, it's probably something that we all know, but I think sometimes we forget when we're doing drug studies or interpreting drug studies, you know, in the neuromodulatory system, you know, it's never one thing working on its own, and these are also interrelated. I mean, even the synthesis of noradrenaline and dopamine is you know, they're sort of steps of the same process. And I think if you modulate any sort of these neuromodulators, you kind of have to think about it as, you know, if you're trying to work on one face of a Rubik's Cube, but we're always changing the others at the same time. So you should never think of pharmacological manipulations as clean. But yeah, the good thing is useful, I guess, for sort of interpretation and thinking is that while it does raise dopamine throughout the whole brain, it doesn't really affect dopamine levels in the striatum, and that's because the noradrenaline reuptaker, the noradrenaline reuptake inhibitor doesn't sort of work on dopamine in the striatum. So you kind of get this cortical raise in dopamine, but not in the striatum. And so, yeah, coming back to then, I guess, the idea of, you know, what cognitive tasks do you kind of throw at this question? You know, if you kind of have symptoms that you care about, you've got a way of measuring the noradrenergic system, you've got a drug that you think might work, and, you know, then you need to kind of capture it in a sort of accurate way, I guess, or as accurate as possible using cognitive tasks. And I guess I sort of told you already just how sort of complex and wide-ranging this the neurogenetic system is. So as part of this study, we tried to sort of, you know, chuck a few, you know, sort of cast a wide net and use, you know, a few different tasks that we thought would kind of, you know, capture different aspects of the sort of complexity of what this system is doing. And I guess to come back to this little plot that I showed you before, the results that I'll walk you through are focusing on apathy, this kind of I guess, yeah, this sort of dimension of apathy or impulsivity, which you can kind of think about as a dimension of engagement with the environment and also learning. So in the apathy and impulsivity, you can, yeah, I mean, yeah, you know, it's, I don't know if it's just, you can see, you know, it's sort of, you know, you can operationalize these things in so many ways. You know, apathy, it's, I guess, you know, motivation is a huge thing, and impulsivity and the kind of, I guess, behaviors that we can think about under that umbrella, they're, you know, they're big topics. But you'll see that we've focused on, two different tasks, which we can capture them quite nicely. And yeah, the spectrum, I don't know. I mean, I don't know if any of you are sort of sitting there thinking, like, why would you think about apathy and impulsivity, which are, you know, people think of two ends of the spectrum, you know, being modulated by the same thing. But the fact is that they are, you know, it's again a kind of nod to the complexity of these systems. It's not like, you know, you can't sort of think, oh yeah, you know, dopamine, that does motivated behavior, noradrenaline, that does inhibited behavior. You know, all of these systems are sort of involved in nuanced way of sort of regulating, I guess, adaptive engagement with the environment. So if the noradrenergic system is sort of driving that, and if it goes awry, it can manifest in, you know, in the same person in both apathy and impulsivity, and certainly in some of the diseases we're interested in, and in healthy people, you do see that apathy and impulsivity correlate. So it does make sense to think of them together, even though, you know, sometimes you kind of think of them at opposite ends of the spectrum. So yeah, we used two tasks to look at apathy and impulsivity. One's a stop signal task that people will be familiar with, I think. But anyway, and then another is this sort of fancy effort-based visual motor task that a clever colleague developed. But I guess the point to sort of note here, and hopefully it's people are sympathetic to it in a cognitive neuroscience context, is that something that we try to do with the behavioural tasks we use is really try to use tasks that we can apply cognitive or behavioural modelling to, so we can get these different parameters that we hope capture behaviour in a slightly more nuanced way. I mean, you know, when you're dealing with a task and the kind of, I guess, usual behavioural output you get, you know, you'll get a sort of reaction time and, you know, whether something was correct or incorrect. But I guess, yeah, the real benefit of sort of cognitive or behavioural modelling is to add a bit more nuance in that it sort of tells you a little bit about, I guess, the kind of strategy or approach behind those raw performance scores. So we try where possible to use tasks that lend themselves to those kind of modelling. Does everyone know what the stop signal task is? I can skip over a few slides here, just in the interest of time. And you get to miss a funny slide of it in action, but take my word, it's funny. But yeah, the stop signal task is it's a simple one, but actually, I don't know, I think it's a good one. We're good because it has beautiful, robust results cross species. The other reason I like it, there's a lot of talk about what a naturalistic task is and we need to use naturalistic tasks, and I'm in full agreement with that. And there's different ways of going about it. In fMRI, they seem to think showing people a movie is a naturalistic task, which again, it's better than rest, but it's not necessarily naturalistic. And there's so many challenges to it. But actually, one way I think about naturalistic tasks is actually tasks that kind of actually capture something that your brain does, in regular life, right? When I think about the NBAC task, right? I don't think I'm ever doing that. I don't think there's any times in my life when I'm, doing something. The thing is, you can make me do it. If you lock me in a scan or in a testing room and tell me to do the NBAC, I can do it. But I don't really think my brain's doing that often. But actually, the stop signal task, this idea of inhibiting a prepotent response, I think that is happening a lot in everyday life. Imagine how often, you're about to say that really mean thing in an argument or sort of step off the curb, and you see a car coming. And I do think there is really something to the fact of tasks that do actually relate to a behavior that our brain actually just does out in the world. And that's why I do think it is a task that has quite a bit of purchase in both in cross species and sort of looking for neural correlates and also in measuring it in disease. But anyway, yes, just to cut a long story short, we used a fancy Bayesian hierarchical estimation of the stop signal reaction time. That's the measure at how well people are inhibiting things. And that was, again, just to take the approach of being able to estimate these parameters, but also trying to use Bayesian hierarchical approaches, which are really good in these sort of smaller samples. I didn't even mention, this is in 19 people with Parkinson's, right? So you can imagine these are small samples. They're older people with an illness, so you're not giving them hundreds of trials. So using hierarchical modeling is very helpful for the kind of extra statistical power it can bring. The other task that we use, and I encourage you to read the paper, it's something you're interested in. It's a lovely task that was developed by a colleague, and it's kind of based on the idea that apathy or motivation, like engagement with the world, might actually relate to this precision you have over your priors. So people that are kind of familiar with the Bayesian way of viewing things, which is not necessarily my field, although I really like this conceptualization. Essentially priors, your kind of beliefs about the world or the state of the world or what you want in the world. And the idea that if you hold these priors with high precision, you've got like a clear idea of sort of how you want your environment to look or what you want, right? Like the dog, he's got a really clear idea that he wants a bone. If you have that kind of precise prior, you'll actually, and if you don't have the bone in front of you, if your world isn't looking how you want it, you'll actually actively engage to resolve that, right? So you'll kind of engage with your environment, you'll do stuff to get the thing that you want, right? If you have imprecise priors, like you got a vague idea, it's like, I might want a bone, might want this, might want this. And then if your environment doesn't look how you want, you'll actually passively revise your priors. So you won't, instead of kind of getting up to get the thing you want, you'll just sort of, you've got that weak belief about what you want, and you'll just kind of, oh yeah, man, I'll just change that instead of actively revising it. So the idea behind that is that it's sort of this sort of imprecise priors, I guess, kind of have this effect in these illnesses. If we think about it, and it's only one way to think about apathy in these illnesses, of course, but it sort of equates to, I guess, disengagement with the world, because you're not actively resolving these kind of inconsistency between your model of the world and what the actual world looks like. So I think it's a nice way of thinking about it. And I will skip over the details because of course running out of time. But it's a lovely task that you can check out. It's a visual motor effort task, and essentially through lots of fancy pants Bayesian mathematics, you can sort of use the kind of people's performance error on this task, and then their error of sort of estimation of where they think the ball would land. You can use that to sort of infer a prior weighting. So sort of how precise or how much weight people are putting on their priors. And as part of this study and other work, Frank was able to show that, even just linking it to sort of an apathy scale in people with Parkinson's, you do see a relationship where people with higher apathy have sort of a lower, less precise priors, we can say. So all that kind of faffing around about tasks, we get to here where you can see that, we've got these kind of two tasks, one that measuring sort of, apathy, one measuring impulsivity. And yeah, the question actually, there's of course lots of results in these studies. But actually the key question that we really cared about, if we were thinking about kind of does the locus coeruleus kind of predict how people respond when we give them a drug, essentially comes down to a very sort of simple equation of whether there's, of whether drug status, so whether people are on the drug or on placebo, interacts with their LCCNR, which is their LC integrity, right? And in both of these studies where we publish these, we see a significant interaction. And this is, of course, a very ugly plot that you're not able to really get what we mean. But this plots it sort of more clearly. So you can see when we think about the stop signal reaction time class. So people, when we gave them the drug, people who got better, they were better able at inhibiting their responses. They were people with a more impaired locus coeruleus, right? Similarly, on the apathy task, the people who got better, they sharpened their priors as part of their task when we gave them the drug. They had a more impaired locus coeruleus. So basically what this is telling us is that LC integrity, it did matter for responsivity to the drug in Parkinson's. And in both of these cases, people with more LC degeneration, they did better when we gave them the drug. And so you can kind of think of it, you can sort of imagine if people's locus coeruleus is not altogether that impaired and you give them a drug, maybe it'll do, give them a noradrenergic drug, maybe it'll do nothing, or maybe it'll make them worse, right? If their system is way too impaired, they're kind of almost past the point of where you can help them because the drug has nothing to work on. But you can kind of imagine, I guess, a sweet spot where people have a certain level of impairment in the locus coeruleus noradrenergic system, and you can give them the drug, and they're the people at the level that will be helped when you give them the drug. They'll do better in terms of their behaviour. So you can imagine, of course, this has implications for even sort of clinical trials where you might do stratified selection. You could imagine, okay, we want to do a big trial to show that atomoxetine helps people with Parkinson's. Let's pick the people that have, a certain level of LC degeneration. So automatically you have this stratified sample that you're going to actually find effects in the drug. Because normally, so much of the reason that at least in Parkinson's and Alzheimer's, that people aren't getting these neurogenetic drugs is because you do a trial and, you never get effects because you've got this mixture of people who are, doing better, doing nothing, or doing worse. So even, you can sort of imagine the possibilities for that for clinical trials and personalized medicine. And yeah, with colleagues in Cambridge, they're working on something like that now. And I haven't left much time to tell you about the study and learning. I'll try to finish at 10:02, so I'll just, yeah. And I'll be around like in the afternoon so people can come and chat with me, which will be lovely. Yeah, but yeah, the other sort of thing that we're super interested in looking at this is learning. And yeah, this is just a slide of, basically taking different timescales of how we learn. So stuff that's happening at the millisecond level, that's dominated by, LTP and LTD plasticity and things. And then these kind of longer ways of engaging with the environment. And consolidation over days and months. Noradrenaline is involved in all of these phases of learning. It's very richly involved in learning, and it just doesn't get as much press as dopamine. Like people are, when people think about, when people talk about learning, it's sort of synonymous with dopamine. So if you take away anything from this talk, is noradrenaline is far more interesting and far more important than dopamine. Not that it's a competition, but yeah, as you can see, I've already told you, it does all this great stuff. It's implicated in loads of different sort of ways of thinking about learning. So from the idea of kind of governing explore, exploit, which I already told you about with the different phasic and tonic responses. So governing, sort of whether you're keeping it doing a task versus going and doing more. It's, again, for people that are interested more in more of the kind of, I guess, predictive coding type ideas. It's implicated in these sort of global high-level prediction error signals. So these ideas of sort of updating your models about the world, which I think I've already alluded to. But again, just imagine the system. It can project, to the whole brain. What a great system to help you kind of, oh wow, there's something is different in my world. I need to relearn this. I need to change this, update this model. There's, you can from the at the level of the locus coeruleus. There are prediction error signals, that's been sort of really well replicated in recent years that are projecting to sort of cortical levels, which is super interesting. And again, something that most people just think about in terms of dopamine. And there's actually a few slides here that kind of unpack what it's doing at these different phases, but I'm going to skip over. I'm sorry. You can appreciate that they're nice, but it's basically just saying that the noradrenaline is involved across the different phases of sort of, stimulus response and then responding to feedback. And in Parkinson's, as you probably know, the focus of noradrenaline has really been on the striatal dopaminergic pathway, where medicated and unmedicated patients perform differently in reinforcement learning. And so, of course, being fans of the noradrenergic system, we wanted to look at reinforcement learning in Parkinson's, and we used a very typical reinforcement learning task with gain and loss, which was a probabilistic task. And again, we applied this sort of hierarchical modeling, again, to sort of deal with these small sample sizes and such. And we didn't do anything fancy. We ended up with a classic reinforcement learning rate model where we're looking at the learning rate and the temperature parameter. So the learning rate is kind of how much you sort of update your behavior based on recent feedback or not. And then the temperature, which is particularly interesting. And well, this was the kind of thing that when we decided to look at learning, I really wanted to see if it might modulate the temperature parameter, because this is the one that we relate to exploit this way of kind of, doing, focusing on the thing or changing the thing. So that was sort of my hypothesis of that it might be sensitive to that. And yeah, the group level modeling results, it's their distributions, because we did Bayesian modeling, so you get like these nice little whale looking things. But quite beautifully, you can actually see, even just with this kind of, the sort of behavioral comparison, the people on atomoxetine, for the behavioral, the beta parameter, they look more like controls. So they've moved towards where our controls are, right? And this is quite clear when you look at the subject level parameters. You can see that, again, when people go from placebo to atomoxetine, they're getting closer to where the controls are, who are in blue. So at that, even just at that level of kind of giving them the drug and looking at the behavior with this modeling, of course, we see that the patients when they're on atomoxetine, they perform similar to healthy controls. And we did some simulations to sort of, this is to sort of show in the joint parameter space. So the idea that, you've got a learning rate parameter and temperature parameter, but they're actually, they are interlinked both statistically and I guess, sort of conceptually, right? So one thing I really like to think about is how to look at different parameters in the kind of joint space. So this is essentially a simulation of, what's the optimal combination of an alpha parameter and a beta parameter that gives you the highest hit rate, right? So when we plot the, and sorry that I'm rushing, but hopefully it's clear. But when we plot the people in the joint parameter space, the kind of key thing the key result of this is that when we measured basically the Euclidean distance between the optimal simulations and what the people do on and off the drug, by giving people the drug, it brought them closer to this sort of optimal set point of how you should perform on the task. So yeah, the take home to that is that people on the drug, when we think of their learning strategy, which is, the sort of I think of the kind of these, these sort of learning rate and temperature parameters, it's kind of characterizing how you're engaging the task. It's sort of your strategy in the task. So our patients looked a lot more like controls and sort of healthy normative normative simulations. And very nicely, we had some pupil as well, and on the drug, people's baseline pupil was higher in the atomoxetine. And when we looked at the phasic pupil response, sorry, there's some bars missing here, but essentially we looked at the temporal derivative. This is people's pupil in the feedback period. So when they do the task and they kind of get it right or wrong, and they get sort of the feedback of whether it's right or wrong, we see this increased phasic response under atomoxetine, where the pupil, the phasic pupil response is sort of meant to be a surrogate of phasic response in the locus coeruleus. And so we're seeing an elevated phasic response in our patients when we give them the drug. And actually how they responded on the drug, it did relate to their baseline pupil, with the kind of take home there being that people that sort of had a higher arousal response, so bigger baseline pupil on the drug, they did better behaviorally on the drug. And yeah, so it just, atomoxetine, quite amazingly, really sort of modulated learning strategy in these patients, particularly in the beta parameter, which was quite nice because we were sort of hoping that. And we can talk about lessons learned, you know, in the chat. And I'd actually been quickly flagging just some of the stuff we're doing. I can do this in 1 minute. But yeah, basically we're trying to do this using a different neurogenetic drug called guanfacine, which is a super exciting drug because it's a little bit less messy than noradrenaline. Has these beautiful circuitry that we know about. It modulates the alpha-2A receptor, and we're currently trying to get that up and running in Parkinson's patients, but it's way harder to do in Sydney because it's actually a clinical trial, which is horrible to do, whereas when we did it in Cambridge, it was kind of a nice loophole where it didn't have to be a clinical trial. So it's happening, but slowly. And yeah, just to flag some of the other cool stuff we're doing in the lab. We're really interested in neuromodulatory systems and sleep, and trying to kind of look at the large-scale brain patterns that happen around sleep spindles and trying to link that with neurogenetic function, because there's all this cool work coming out in animal models of how neurogenetic oscillations drive patterns in sleep. And the other really exciting thing we're doing, we were really lucky last year with some great colleagues to get a discovery project where we're trying to look at the neural basis of compositional cognition using cool fMRI tasks and things like that. So yeah, sorry I was a bit long. And yeah, please get in touch if you'd be interested in working with us, because we're, you know, hiring and looking for students and stuff, and yeah, hopefully doing stuff that is kind of cool. Anyway, thank you.
Thank you very much, Claire. I think this talk just shows us that we'll have to invite you again. So to see the results of the.
I'll make less slides next time.
No, this was beautiful. Let's open this room for questions. First, do we have any questions from students? Oh, I see Layla. Yeah.
All right. Hi, thank you so much. That was great. Sorry, I maybe missed one part. So you see the impact of atomoxetine in stop signal and inhibition, and it was so based on the results in the stop signal, the error was less. But when you look at the learning, reinforcement learning, so there's no impact between placebo and atomoxetine group, right? Or I missed that part.
I guess. Yeah, I guess yeah, it probably doesn't make sense to think about the stop signal results and the learning results together in a way. I guess because yeah, in the stop signal results, we actually, I mean, I kind of, you know, buried some of the results, but when we looked at the groups for the stop signal task, the Parkinson's, when they were on atomoxetine and when they were on placebo, we didn't actually see a group difference, right? But where we saw the effect is when we looked at the interaction with the locus coeruleus. So when we did that interaction, we could see that the people who had a more impaired locus coeruleus did better. But the cool thing about the reinforcement learning results is it actually just at the level of behavior, like, and actually I didn't even present it, but even just presenting the hit rates, right, that the groups were really different. And anybody who knows, maybe Tony knows from doing you know, drug studies in mice or humans, you basically never see a behavioral effect. You essentially kind of have to really dig for it. You know, you have to kind of stratify, oh, well, if we look at it in the impulsive rats or the non-impulsive, then we see an effect. But actually just at the group level, like really just seeing that, wow, when we gave the patients these drugs, you know, they looked a lot more like controls. So yeah, it was really one of the kind of first times we've sort of seen kind of such a clear effect without sort of needing to kind of consider, yeah, the baseline dependency or these sort of, you know, like the interaction type effects. Yeah.
Another question.
That was a really great talk. Yeah, thanks. I was just wondering about how you administer the atomoxetine drug. So my understanding of how it works, it's pretty rudimentary, but you usually have to give people a little time to kind of get their noradrenergic kind of tone going. And usually if you take the drug for like a couple of months, say, you'll perform probably better than you would if you just took a single dose. And I was kind of wondering like what your thoughts on about that and how you usually think about these problems.
Yeah, no, it's a really good point, and it's a kind of good point because again, it's sort of, you know, when we think about these sort of single dose drug studies, I do think that they have really good, they have really real potential to be clinically relevant, but they're also just kind of cool to help us like understand how the brain works. And so, you know, when we kind of think about just trying to understand the brain with them, we kind of ignore those effects. But yeah, at the level, I guess you can sort of answer that in kind of two ways, sort of at the level of the kind of the single dose, you know, even just kind of making sure, you know, you administer the drug and you have to wait in atomoxetine in the case of sort of you wait about 2 hours. That's when it's sort of meant to reach its sort of peak in the plasma. Study with guanfacine, because you can only get extended release. We'll be sitting around and waiting a bit longer, so that'll be fun. But yeah, you're absolutely right. The sort of kind of acute effects of a drug can be quite different. I mean, I think we know a lot about that in SSRIs because people have done a lot of, and even just sort of anecdotally, the idea that, people start on an antidepressant, that it takes, a couple of weeks for it to actually kick in and make them feel better. So certainly there would be different effects long term. I don't think we know as much about it, but essentially, and I guess, I didn't sort of make it explicit, but essentially, I feel like if we imagine the kind of clinical application of these single dose studies, I think they're a wonderful nod to possibilities, but until you actually do it out in the real world and give people the drug, every day for 8 weeks, you're never going to know. And that's actually, I skipped past it, but the colleagues that I worked with in Cambridge on this, in another disease, it's similar to Parkinson's, progressive supranuclear palsy, or PSP. They're currently running a longer term clinical trial with atomoxetine. And even though they've not stratified people on the locus coeruleus integrity, they are measuring the locus coeruleus, like we said, and they're doing the subsignal task. So that will kind of tie in nicely. But yeah, that's the proof in the pudding, if it actually helps, every day.
Thanks, Claire. Lovely work. I was wondering, with your degeneration of the LC in correlation with apathy and impulsivity, have you measured other brain areas? Could the correlation actually be with other brain areas that are co-degenerating in those brains?
Yeah. So in the subsignal paper, actually, a very astute reviewer asked a similar question, and we did, and so we actually took. We used the same approach to to get the kind of contrast of the catecholaminergic of the dopaminergic nuclei in the substantia nigra. So we got that same measure of that integrity, and then we also took, I think, a whole brain measure. So it's a little bit cursory, but yeah, they didn't have they didn't sort of, I guess, explain the behavioural response in the same way. So that was, I guess, a way to kind of one-placate the reviewer and to sort of chase down that question. But I sort of take your point in a way that, it is an oversimplification to think, it's just going to be this area. I guess the way that we think about it is, the integrity of the LC is likely to be sort of some kind of surrogate of the whole system, So I think it should relate. And actually it's ridiculous. Like, we're still mining through this data. We had kind of collected a lot of data, but some of what we did in the baseline scanning was even just to collect resting state. So at the moment, we're having a little look about because it's at 70, we can actually sort of look at locus coeruleus kind of projections throughout the brain, and even just to kind of actually answer that question of whether does the LC integrity, as I've presented here, is that would we see the same thing if we looked at sort of measures of kind of functional LC prediction or projection to the rest of the brain? So it's something we're chasing up, and definitely an oversimplification. But I would say a pragmatic oversimplification if you imagine the kind of possibility in clinical trials, right? The idea of, I've presented very fancy 7T scanning, but these neuromelanin sensitive sequences for the LC, they can be done at 3T, and you don't necessarily need a fancy scanner. They can be done in, 7 minutes. So if you imagine kind of like, let's screen someone for a trial, pop them in a scanner, a quick scan, and whereas, doing something like some, fancy, complex sort of functional analysis. So yeah, I guess it's a simplification that I do think could be translatable to clinical trials and even, personalized medicine at some point, maybe.
Could it be a surrogate for how much and what dose of L-DOPA they've had previously? Do you have medication data prior to testing?
Yeah, we do. And we looked at sort of different correlations with that and didn't find it. But again, it's a small sample, and but again, it's funny. I mean, it's tricky when you bring in sort of medication dose in Parkinson's. Inevitably it's going to sort of correlate with other disease measures and things like that. But the way I think about it, and again, this would, it's sort of, I've presented kind of the LC as, being, impaired in Parkinson's, and, it generally is. But, in these diseases, they're so complex. There's so much heterogeneity. You know, you can see two patients with Parkinson's or with Alzheimer's and Huntington's as well, who are apples and oranges. They look completely different. And we know that there's so many different phenotypes you can characterize them on in different ways. And you know, I've no doubt that you know, if we were to characterize them at the level of, I guess, you know, catecholaminergic change, you'd have people that are more sort of dopamine sort of driven change versus more noradrenergic. And there is some ideas kind of emerging of sort of a noradrenergic subtype of Parkinson's. So yeah, I think there would be relationships with medication, but yeah, I think people will sort of end up falling into kind of two camps in relation to dopamine and noradrenaline and Parkinson's.
In single dose atomoxetine, do they know whether they've had that versus placebo? Is it totally
No, so
blind?
it was blind. Yeah. So
They don't report whether they
no,
knew.
and we did, you know, we do like the little, the sort of visual analog scales to kind of measure if people change. And yeah, anecdote, I mean, nobody sort of got unwell in it. None of the patients sort of said that they knew. And this is actually, you know, part of the reason why atomoxetine, I think, has a lot of potential in these illnesses is because it's a reuptake. It's not an agonist, so it's a reuptake inhibitor, so it's gentler, you know. And this, I mean, you know, it might actually knock people off their feet. We're a little bit worried, but there's a trial at the moment we've seen in people